Thank you, and thanks very much for having me. And as the title suggests, we were looking at variants in coronary artery disease, and we wanted to understand better what may be the epigenetic consequences in this. And as a very, very high-level introduction to coronary artery disease, it's the most common cardiovascular disease, also the most common cause of death, and it's characterized by atherosclerosis in the arteries of the heart, which reduces the blood flow, can lead to thrombosis, and thus to a heart infarct. And it's known to be a polygenic disease. And there have been different GWAS studies that try to understand what's happening there on a genetic level. And one which I want to particularly highlight is a study from 2022 where they combined a lot of data and in the end had over 1 million participants in the study with over 200,000 cases. So it's a very rich data set to find variants associated with coronary artery disease. And they actually already did a lot of different strategies to find genes which could explain this association. So I won't name them all, but just as illustration was closest to variant or drug target or EQTL data. But we thought that there is a certain bias towards protein coding genes. And also we thought that maybe it's worth to look also a bit more into the non-coding regions. And I will come back to this later. But first, I mean, this was already touched upon that it's worth to look at consequences also in non-coding regions, but to show this also from studies where they summarized this. So this is data where they collected lots of different GWAS studies across all kinds of traits, and just were looking where these variants actually located. And we see that the majority is in introns and also intergenic regions. And if you then check if the chromatin is active at these positions, you also find an enrichment. So we can say, because we have so many different variants across all traits that are located in non-coding regions, it's kind of worth to look at this and not neglect this information. And I was telling you that we wanted to look at epigenetic consequences or consequences in gene regulation. What do I mean by this? We were specifically looking at binding of transcription factors. So you have a gene, you have the promoter, and transcription factors can bind there. And then you can have enhancer regions, which can be quite far away. And transcription factors can also bind there and then interact with the other ones and then influence gene transcription. And this binding is happening in a sequence-specific fashion. So these TFs, they recognize certain motifs in the sequence. And because this is sequence specific, and now we have variants which change the sequence, we can ask whether this has a consequence for the binding. And to analyze this, we have a tool called SNEAP, developed by my colleague Nina Baumgarten. And this is comparing then the wild type sequence to the mutated sequence. So we exchange one base, and we're asking whether this exchange of the base is affecting the transcription factor binding, because you might destroy the motif. But you could also have a gain of a transcription factor binding site. And we ran this tool across the whole GWAS study I showed you earlier with over 1 million participants. So we knew for the variants associated to coronary artery disease, do they affect transcription factor binding or not? And also whether it's a gain or is it a loss? And this was the first type of information we needed. But we also wanted to know where could these transcription factors actually bind, because they do so in active or open chromatin regions. So they just don't bind anywhere. And this type of information is highly cell type specific. So what we did is we collected epigenome data across 45 different cell types, all assays that are kind of associated to open chromatin. And we had data from hematopoietic cells, immune cells, endothelial cells, and more. So in total 45, as I said. But there's one more information that we were missing, because in the end we wanted to get to the genes, but we were still missing the information which genes actually, which genes do these enhancers regulate. So we have the enhancer information, but we needed to get to the gene. And for this, we used an implementation of the activity by contact score, which just very rough introduction. It uses enhancer activity data and chromatin contact data to predict the target genes of the enhancers. And then we actually have everything together to get to the candidate genes, because we have the variants from the GWAS studies. We know whether they affect transcription factor binding. We know where the transcription factors could bind in a cell-type-specific fashion. We know where these enhancers then map to genes, and then this brings us to the candidate genes, because their regulation might be affected by the variants. To give you an illustration of the data, we had in total almost 1,600 candidates, which is quite a lot. And we had around 50% which are already associated to coronary artery disease in one way or another. And what I'm showing you here is just the top 20. We have it, of course, for all. It's just for illustration. We have on the y-axis, we have the top 20 genes. And on the x-axis, you can see all the different cell types for which we had data for. And what we can now do with this data set is we can ask for each gene in which cell type, how many enhancers of this gene have changes in transcription factor binding. And the hypothesis is that the more of the regulation of a gene is affected, the more likely it could be involved in the disease. And we layered this kind of table or this information with a lot of additional data. We looked at co-expression with known genes. We looked at other analysis like co-localization with EQTL data and literature evidence. And yeah, this is available for all the genes, as I said. And another thing that we did is we wanted to see if we can kind of bridge these genes that we find to the coronary artery disease by doing a phenome-wide association study. And for this, we took our candidate genes, and then we took the EQTLs from different resources like GTEx, because we know that these EQTLs are associated to the gene. And then we were doing a co-localization analysis with different GWAS traits to see if the EQTLs of these genes are associated to traits which could make sense for coronary artery disease. And we found for almost 80% of the genes, we found an association with any of these shown traits. This is the left histogram first, and we find a lot of co-localization of the EQTLs with variants involved in inflammation, with lipids or blood pressure. And because EQTL data is tissue-specific, we can also ask in which tissues do we find how many associations, and the highest number of associations we find in the arteries, but also in adipose tissue. Which is also nice about this data set, because we can also ask from the perspective of the transcription factors, because we can ask whether certain transcription factors are more often affected than you would expect just by chance. And here I'm showing you only the ones that have an odds ratio of higher than 5, so 5 times more likely to have a change in the binding size than you would expect with random SNPs. And also we have this information on a cell type specific level, and you see that some are kind of broadly affected across many cell types, and these AP1 factors, for example, are kind of specific in adipose tissues, which was a little surprising to see. And we also checked whether these transcription factors are differentially expressed in patient data, and this was actually the case also for the vast majority of those. So you find these TFs differentially expressed in tissues of patients with coronary artery disease. And this actually already brings me to my summary, so what I showed you today. And so we started with the GWAS study. We looked for enhancers. We looked whether they affect transcription factor binding. I showed you our list of candidate disease genes, the PHEWAS analysis that we did, the affected transcription factors. What I didn't show you was that there was also functional validation of a long non-coding RNA, it's IQCH-AS1. But if you want to look up more of this, I would kindly redirect you to our preprint. And with this, thank you very much to my co-authors, and thank you for your attention, and happy to take questions.
And while we take questions, please, the next speaker come to stage. Yes.
That was very interesting. Yes, that is on. Sorry. That was very interesting. Thank you very much. I'm curious about the story at the end where you showed the adiposity or the genes linked to adiposity and the enrichment of transcription factors in adipose tissue. Do you think you're actually just picking up the fact that people who have very high BMI might have a very high risk of CAD? So it's not quite that specific to the disease per se, but rather it's the risk factor for the disease.
Yeah, I think we're finding a lot which is just going via the risks, but I think this also kind of tricky with these types of studies, because if you compare, patients with controls, you have the people with the risk factors also in your patient cohort. So yeah, it's, for example, the functional validation, it was also then done in adipose tissues, and it looks like this one is also protective, but also indirect via these lipid metabolism. So that is kind of reducing the circulating lipids, and this might be then also kind of a protective factor for coronary artery disease.
Thank you. We are running a bit of time, so thank you. And next speaker.